Hi, everyone. Thanks, Armando. And I also want to thank the organizers of this symposium for inviting me and letting me explain some of the results I have done with the atomic force microscopy. While I'm working now at the Facultad de Ciencia e Ingeniería en Alimentos at the Universidad Tecnica de Ambato. And here we have, finally, we have an AFM, but it's due to the huge effort of the Dirección de Investigación y Desarrollo from this university, and also from the Facultad, and also from other companies like Incolor, for example. And finally, we get it, but we have just get it recently. So I didn't have much time to develop so many results to this talk. So what I'm going to show to you today is some of the results I have obtained in a previous collaboration I had made in the nanoscience in Madrid, Spain. So the title of the talk is In Exploring Biostructures at the bottom. And why this title? This is because I think that the biostructures has a lot to tell to us. Nature, in a certain way, looks for some strategies to overcome the same difficulties that the very same nature is imposing to us. So exploring how these structures, how these strategies have done with the evolution can help us to do some useful network for us. So this is the outline of the talk. I will start talking why it's important to use the atomic force microscopy to this particular field of biostructures. I will talk a little bit about the capabilities that we have now at Universidad Tecnica de Ambato. And later I will afterwards talk about some of the results I have obtained. Of course, it's not a long journey. I have collaborating with plenty of people from different teams, experts in biology, in food, in protein scaffoldings, in biophysics, in nanomedicine. And later I will focus on one particular study that is related to how to measure the extracellular vesicles released from living cells, so in vivo. After that, I will skip a little bit about biology and turn into some field that is in a certain way related to biology, but not directly. That is the nanotribology, that is the friction at the nanoscale. And I will talk a little bit about a study about how to use the Violin as this nanotribological instrument. So as I think most of you know, the FM uses a very sharp tip to detect the forces that is experienced with between the tip and the sample. And if you think it's quite easy. I mean, if you are in a dark room, you don't have light at all, what do? You can fall off. But apart from that, what do? You touch. So that's exactly what we are doing. If we want to check how are the structures at the nanoscale, and we cannot see because basically the light interferes with itself at this scale. So we cannot see that, what we do with that. There are different modes, as you know, but basically we are looking for different forces that affects the tip by the surface of the sample. And there's another important thing that the AFM provides is the control of the force. By this, we can measure, we can touch these samples with an extremely control of the force that the tip is applying on the sample. So this opens the possibility to measure some very soft samples like biological ones. So for example, here I want to tell that This is the AFM we have now at the Universidad Tecnica de Ambato from Park Systems, is XE7. And we hope to continue some collaboration and open other ones to explore some of the possibilities that the biodiversity that this country has can be checked by this technique. So, for example, what can we measure with FM? I have, for instance, checked how is the peptidoglycan network of the sacculi of some bacteria. For example, the Escherichia coli, most of the bacteria have some structure that maintains the shape and prevents them to break up, basically. And one of the strategies is to form this peptidoglycan network that can be checked by AFM, just by depositing, adhering this sacculi on a flat surface. I think, if I remember, no, it was mica, not HOPG. We can do that by HOPG, but it was done on mica. And we can check how this peptidoglycan network is affected by the media, because this is something that I haven't told you, but probably you know. We can measure with some media around it, in the surroundings. So we can check the media, and in vivo, in real time, we can see how it is affected, the sample, by this media. So, for example, in this example here, this other experiment that we did, these are filaments made of FTSZ. This is a protein that is one of the plethora of proteins that is forming this septum that helps to divide the bacteria. There are plenty of other ones, but this is one of them. So by putting that protein on a flat surface, it is forming these polymers. But this is amazing that the polymers are not have not formed, and how to say, they stay in this shape. No, they are. It's a dynamic combination. So in the media, there are proteins, there are FTSZ molecules, and they interchange with these polymers. So it is a dynamic polymerization. It's quite difficult to measure that with other techniques. So this is possible with the FM. Another example is what I'm presenting here. This is a virus. This is an IBDV virus. It's not coronavirus, something like that. It affects only the fishes. And we wanted to know what's the effect of RNA on the capsid of this virus. Of course, RNA is involved in this reproducing issue, but apart from that, what if it is doing something else? It is helping, we discovered that it is helping to the capsid of the virus to maintain this morphology. So in the upper line, we press the capsid with tip in a controlled way, and we saw that it is broke up quite easily. So once we have a hole, it breaks up. And this is because in the inside, there is not a lot of RNA. But if we increase the amount of RNA inside, you can see that with the same force, the capsid can bear much more force. It can remain with the shape a certain way. So it's helping also for the scaffolding of this capsid. Here in the upper part, in the upper right part, I'm showing one of the few results that I have done so far at the Universidad Tecnica de Ambato. It was made completely in this lab. It is a film formed by banana, and trying to reuse the waste of this material, we can make some edible films, and some of them were more breakable than others. how can we measure that? So by measuring at the surface with AFM, we can see that one has some holes on it, and the other one has less. So probably this is the reason of the elastic properties of plastic properties, I would say. What else can we do? We can make the proteins do what we want they to do. We can manage these kind of proteins, CTPR, and for example, we can cover a surface and we can coat it completely. And also we can, this is a recent study, we can put some beads, some metallic beads on it. So we can not only cover that, but make it metallic. For example, this mica can be changed to metallic with metallic properties, at least on the surface. Here, what we did is to put the same proteins, but in a certain way that we can make these furrows with optical properties. That could be interesting. And also we can see what nature has done. This is an experiment in where we look for differences in a bilayer. We deposit a bilayer of lipids, and we saw how it is affected by proteins, how it is affected by other molecules that can be incorporated inside, for example, catechins or cholesterol. There's a change in the bilayer, of course, and we can explore how is this modification by using the AFM in a media, of course. Here we can just see how is the morphology of RNA, G quadruplexes, DNA, a lot of nucleic acids. Here we can see directly the bacteria. We can check if there are some bacteria and some flakes of HOPG or a few layer graphene, I would say. And we can see if the bacteria are alive in the green color or dead in the red one. This is for batteries, very ecological batteries. And we can also check how are the structures of some flakes of some fishes. These nanostructures may help these fish, for the flake fish, for the elastic properties, but also I think that it is involved in the change in the regime of the liquid that passes very close to this flake. So probably the fish uses this nanostructure to swim much better. Here we wanted to know what's happening to some cells that are deposited on a surface, but the surface has some nanostructure. There is this double arrow is because there is furrows, these grooves that goes in this direction, and we just wanted to see if it affects the cell or not. And what we experienced surprisingly is that the cell with this philopodia is going along these furrows, but also perpendicular to these furrows, to these grooves. And why is that? Rethinking about that, we discovered that it's because they follow the lines that helps the cell to get more attached to the surface. And there are two ways. One is perpendicular because there are more points of contact, and also along the furrows. That was surprising. We can also check by changing the media if these erythrocyte membranes are affected by the ATP addition to the media or not. We can see the actin filaments with the ATP, and of course not without ATP. This is a comparison in between some structures that is formed in the stroma of our eyes after being modified by some femtosecond lasers. There are chirurgical issues for correction of the eyes, and that uses femtosecond lasers. But how they affect the stroma of the eyes? All of them works, but which one was, in which way they works? This is how something that we can check with the FM, and we can compare the stroma affected by different lasers. I don't know what's better. If it is better that it's rough because it links better, stronger, or it is better if it is flatter. This is something that we have to study beyond that. And I'm going to focus now on a study that I think is quite interesting and where AFM has a lot to do. This is how to measure the extracellular vesicles that comes out of the cell. There is a connection in between cells. If there is a virus invasion, for example, now that we have this pandemic, the one cell tells the other one how that there is a danger, so to be prepared. There are plenty of different communication, but this is one of them, and one of them that the exosomes probably helps to overcome the brain blood barrier. So this is quite important. So what we did is to put these cells on a surface, measure it with the FM, just like that. I mean, we need a buffer because we have to maintain the acidity, because we don't have a CO2 system, etc. But just by doing that and measuring the surface, we can see, with a very controlled force, of course, we can see in the protrusions and the pits that are experienced on the surface of the cell. But what's important is that the cell is alive. So the cell was moving while I was doing the experiment with the FM. We can follow some of these protrusions on the surface, some of the tips that are, the pits that are formed. So we can see what the cell is doing. The thing is very surprising. Of course, we can fix it with aldehydes, and we can check that, for example, metallizing the surface with the electron microscopy. We can study the roughness of this the morphology of these extracellular vesicles by depositing them on the surface and fixing them. And also we can change the medium, put some hormones, for example, progesterone, that was the case, on the media, and see that, well, it has some more activity with progesterone. We could check that just by measuring the amount of protrusions in comparison with the addition or not of the hormone. And now I'm going to change a little bit to this field, this nanotribology, because this FM also opens the possibility to measure how friction is working at this scale. This is one of the few techniques that allows us to do that. For example, we can see that at a certain point we can change from a static to a plastic regime. So basically we break the material, the surface of the material. It's a very soft material made of polymers. We can see how much force is needed to push a particle along these furrows and change to another furrow. And also we can play to the nano football, and we can push the particles that are on a flat surface. And we can study how it is turned. But what's important here is that this tilt, this line here, that is the result of the interaction between the tip and the particle, has an angle, and this angle is related to the friction. So friction is acting in a different way as we used to see at the macro scale. In the macro scale, we have basically three kind of frictions: the static friction, the dynamic friction, and also the stick-slip friction that is changing in between the previous two ones. Here, just this is the last study I want to show. Here, I ask this: how is some hair of a bow of a violin affecting the sound? Why some hairs are producing a sound and another hairs not? Of course, it is related to stick-slip friction, but how? So I had a bad violin. I don't have here, so don't worry. But I had a very bad violin, cheap one, and I also have the bow hair of people from the Philharmonic Orchestra. So I compare both. Curiously, the one I have, the bad one, is synthetic, and the one at the Philharmonic Orchestra is natural. So directly related to biology, but it's natural. It's the hair of the tail of a horse. That's it. But it is much better. The sound that gets from that is much better. Using the same string, using the same violin, everything the same, just changing the hair. Why is that? So we discovered with the use of the AFM that the structure is maintained with some periodicity and is less breakable at this scale. So the sound can be much better. I discovered also that the commensurability in between the nanostructure or microstructure of the string and the bow hair is very important for the sound. With the fast Fourier transform, we could see that there are some periodicity in one case and not in the other one. But if we put the rosin, for the bow hair, we have to put the rosin, and if we do that, this was very difficult because the tip gets a stick to this rosin. But finally, I got the images, and I discovered that the periodicity is the same. So by putting the rosin, we have the same thing basically. But you see that if you compare, there are much more rosin in the case of the bad bow hair. So, and how is that related to biology? I think it is very related, but we have plenty of things to discover here in connection between nanotribology and biology, and hopefully I will continue with these experiments. I want to finish by thanking all the teams that have helped me during these plenty of years. I want to thank, of course, Indian Nanociencia Institute, the Facultad de Ciencia Ingeniería de Alimentos, Universidad Tecnica de Ambato, Park Systems for the invitation, and you for your attention. Thank you very much.